Collaboration between genes and the environment on nearsightedness. I've been interested in nearsightedness since I was in 3rd grade and had to wear eyeglasses for the first time. As I grew up, my prescription got stronger and stronger. It was no shock that I had nearsightedness at such an early age because both my parents have eyeglasses that are extremely thick. My research is focused on whether there would be a way to prevent someone like myself, who is genetically determined to have myopia, which is a fancy name for nearsightedness, from being myopic. The eye works in a collaborative manner to make objects in the world come into focus. The optical components of the eye are the lens and the cornea, and together they focus the image of objects in the world onto the retina. This is a picture of an eyeball that is hyperopic or farsighted. When humans are born, their eyes are too short, so the image forms behind the retina. Although normally, when people are hyperopic, they can see image in focus when they are far away, children can see objects in focus because there are muscles that change the shape of the lens in order to bring objects into focus. This is called accommodation. Throughout childhood, the eye grows from front to back or the axial direction. When the length is optimal for the optical components, the image of objects in the world are focused on the retina, and the eye is said to be emmetropic, and it normally stops growing. If the eye continues to grow beyond the point of emmetropic, or beyond the emmetropic point, the person becomes nearsighted or myopic. Myopia has become a worldwide health problem, with more people affected every year. Having myopia makes people more susceptible to other vision-threatening eye problems, such as glaucoma and retinal detachments. Understanding the causes of myopia is important for developing ways to prevent it. The purpose of this study is to evaluate the interaction between genetic and environmental risk factors in myopia. The retina is a tissue in the back of the eye that is made up of many different neurons. Light rays from objects in the world pass through the lens and the cornea and form an image of the object on the retina. Neurons that make up the retina respond to light in dark edges and send information to the brain about the image. For someone with normal vision, signals in their eye tell it to stop growing once the eye grows slightly past the point at which distant objects are in perfect focus. In the modern world, environmental factors such as close work, like playing video games or reading, and exposure to artificial lighting, which contains more red and orange light compared to more blue and green sunlight, confuses the signal and causes the eye to keep growing. This means more time spent outdoors and less time spent doing close work has potential to prevent myopia. There are 3 types of cone photoreceptors, called blue, red, and green cones, which are cells in the retina that allow people to see in fine detail. The genetic risk factor for myopia comes from genetic variation in the photopigments, which are molecules that absorb light in the cone photoreceptors. Photopigments are made of 2 parts. One part is a protein, and the other is a derivative of vitamin A. Combinations of variants in the amino acid sequences of the red and green photopigments make certain individuals more susceptible to myopia and cause their myopia to get worse when exposed to environmental risk factors. This is an image of photoreceptors. When the image is blurred, light from an image is spread more evenly across the photoreceptors. Therefore, neighboring photoreceptors make similar responses. When an image is sharply focused on the retina, light and dark regions of the image can fall between neighboring photoreceptors, so the photoreceptors each make very different responses. Once the eye gets slightly too long and the image is slightly blurred, emmetropization should be complete, and the eye should stop growing. This research project compares the amount of myopia in a group of people who live in Seattle and are exposed to environmental risk factors in the modern world to a group of people who live in a remote area of Argentina where there is almost no access to artificial light and where people live outdoors. The hypothesis is that cone photopigment variants are the major genetic risk factor for developing myopia, and the environment of the modern world, such as in Seattle, allows people to fulfill their genetic predisposition. This hypothesis predicts that people in Seattle will have the full amount of myopia predicted by their amino acid sequences in their photopigments, but people living in remote villages of Argentina who live outdoors and have almost no artificial light will have less myopia than would be predicted from their cone photopigment variants. Experiments involving human subjects were conducted in accordance with the principles embodied in the Declaration of Helsinki. To be enrolled in the study, subjects were required to pass the Richmond HRR color vision test because the cone photopigment genes that are involved in nearsightedness are missing in people who have a red-green color vision defect. DNA was isolated and genotyped for both people of Argentina and Seattle to compare sequences in the red and green photopigments. Also, the refractive error was taken using multiple methods. It is measured in diopters. A refractive error of 0 diopters means the eye is the perfect length for the optics and is imetropic. A positive refractive error means the eye is too short for the optics and the person is farsighted. A negative refractive error means the eye is too long for the optics and the person is nearsighted or myopic. The smaller the number, the more myopic the person is. For example, someone with a refractive error of 2 diopters is more myopic than a person with a refractive error of -1 diopters. Both of the genotyping and refractive error were used to compare the Argentinians and the Seattleites. The gene sequences can predict the genetic susceptibility to myopia, and the refractive error quantifies the myopia. This table was taken from a previous study where DNA was sequenced and the average SER, or myopic potential, was taken from many people with the same amino acid sequences. I used this data to predict the myopic potential of the subjects in my study by matching the amino acid sequences of subjects in this table to subjects in my study. The variant code column will show up in the next couple of tables. It helps keep track of amino acid sequences that are identical. This table shows the data collected from the Seattleites. The columns labeled red and green variant show their actual amino acid sequences in their red and green photopigments. The column labeled SER phocometer is the measurement in diopters of how myopic they are, and the final column labeled predicted SER is their myopic potential based on their amino acid sequences in the second and third columns. The total amount of myopia that the subjects have is close to their myopic potential. This table shows the data collected from the Argentinians. It is set up in the same way the previous table was set up. In contrast to the Seattleites, the total amount of myopia the subjects have is 0.42 times less than their genes would predict. These results support the idea that the specific combination of red and green cone photopigment variants a person has represents a major genetic risk factor for developing myopia. In addition, the results suggest that the environment experienced by people living in artificial environments, such as in buildings with artificial lighting and exposure to books, television, and computers, conspire with the photopigment variants to cause people's eyes to grow too long. In contrast, living in a more natural environment without exposure to modern conveniences can protect a person from getting myopia, even if the person has a high genetic risk factor for myopia in their cone photopigments. These results suggest that it should be possible to devise ways to protect people who live in myopia-causing environments from getting myopia by altering the environment or the way that the environment acts on the cone photoreceptors.